Welcome to the PubMed Daily Review for April 26, 2026. Today we scanned 3,854 new open access papers. Here are the top 5 selected for detailed analysis. Up next, our first paper out of 5, Genome-scale functional mapping of a mammalian whole brain with in vivo Perturb-seq by CRISPR and Xing. Let's break down its key contributions. The brain is incredibly complex, and understanding how individual genes contribute to its function is crucial for tackling neurological and psychiatric disorders. While we've learned a lot from lab-grown cells, we need to see how genes behave in the living brain. This research pioneers a new way to map gene function across the entire mouse brain, opening doors to new insights. To achieve this, the researchers developed an advanced in vivo Perturb-seq method. They systematically knocked out nearly 2,000 disease-associated genes and then analyzed the resulting changes in gene expression across over 7.7 million individual cells. This allowed them to map these functional changes across different brain regions and specific types of neurons. The results are remarkable, providing a functional atlas of nearly 2,000 genes across millions of brain cells. They discovered that gene function is highly specific to cell type, with even closely related genes sometimes having opposite effects on cellular programs. This detailed mapping offers unprecedented insights into the genetic underpinnings of brain function and disease. This work is a major leap forward, offering a detailed map of gene function directly within the living brain. It highlights the critical importance of cell type specificity in understanding brain disorders and opens exciting new avenues for developing precision genetic medicines tailored to specific brain cell populations. Up next, our second paper out of 5, Advances in CRISPR base editing. From molecular evolution to therapeutic applications in genomic medicine by Melik Alishyaslin and Salih Samlioglu from Department of Gene and Cell Therapy, Faculty of Medicine, Akdeniz University, Antalya, Turkey. Let's break down its key contributions. The ability to precisely alter our genetic code has been a long-standing goal in medicine. While CRISPR-Cas9 opened many doors, its reliance on DNA double-strand breaks and inefficient repair pathways presented challenges for certain applications. This led to the invention of base editing, a powerful new tool that allows for direct, targeted single nucleotide changes without breaking the DNA double helix. Base editors are sophisticated molecular machines. They combine a modified CRISPR-Cas9 protein, which acts as a guide to the target DNA, with a deaminase enzyme. This deaminase directly converts one DNA base to another, like changing a C to a T or an A to a G. Crucially, this happens without cutting the DNA double helix, and it doesn't rely on the cell's often inefficient repair mechanisms. This makes base editing a much more precise and efficient way to correct single-letter errors in our genome. The evolution of base editors has been remarkable. Cytosine base editors, CBEs, saw significant efficiency gains, and the development of adenine base editors, ABEs, allowed for the conversion of AT to GC pairs. Together, these editors can now achieve all 4 types of transition mutations. Importantly, they do this with very low levels of unwanted insertions or deletions, typically less than 0.1%. This precision and efficiency have led to successful preclinical studies and the initiation of clinical trials for serious genetic diseases. Base editing represents a significant leap forward in our ability to precisely correct genetic defects. Its reduced reliance on DNA breaks and HDR makes it a potentially safer and more efficient therapeutic strategy. While challenges like efficient delivery to target tissues and minimizing any off-target edits still need to be addressed, the ongoing advancements in base editing technology hold immense promise for treating a wide range of genetic diseases and advancing genomic medicine. Up next, our third paper out of 5, Endothelial NAD plus depletion drives vascular senescence and neuroinflammation via mtDNA cGAS/STING-CD38 signaling in Alzheimer's disease by Qinghua Luo and Xing Zhu from Department of Neurology, the second affiliated hospital, Jiangxi Medical College, Nanchang University, Nanchang, Jiangxi, PR, China. Let's break down its key contributions. Alzheimer's disease is a devastating condition, and while we focused on amyloid and tau, it's becoming clear that problems with blood vessels play a big role, especially as we age. Brain endothelial cells, which form the blood-brain barrier, seem particularly vulnerable. We know that when cells get old and senescent, they can cause inflammation. But what triggers this in the brain's blood vessels in AD? And could NAD+, a vital molecule that declines with age, be involved? To investigate this, the researchers used a mouse model of Alzheimer's disease, the AP/PS1 mice, along with isolated brain endothelial cells and microglia. They explored the molecular pathways involved, using techniques like NAD+ supplementation with nicotinamide riboside, genetic analysis, and various biochemical and imaging methods. Their core idea was to see if amyloid beta triggers NAD+ depletion, leading to a cascade of events that cause cellular aging and inflammation in the blood vessels, and if restoring NAD+ could reverse this. The study found that in the AD mice, amyloid beta indeed led to NAD+ depletion in brain endothelial cells. This triggered a chain reaction involving mitochondrial DNA release and the cGAS STING pathway, promoting cellular senescence and inflammation. Crucially, when they supplemented with nicotinamide riboside to boost NAD+ levels, they saw significant improvements. This treatment suppressed the inflammatory signaling, reduced neuroinflammation, and importantly, restored vascular function and cognitive performance in the mice. So, what does this mean for Alzheimer's disease? This research strongly suggests that boosting NAD+ levels in the brain's blood vessels could be a novel therapeutic approach. By targeting this NAD+ dependent pathway, we might be able to combat the vascular aging and neuroinflammation that contribute to AD. While these are exciting findings in animal models, more research is needed to see if these benefits translate to humans, especially in the early stages of the disease. Up next, our fourth paper out of 5, blocking RNA translation without altering repeat RNAs rescues C9ORF72-related ALS and FTD phenotypes by Xian Jiang and Clotilde Leguerin from Department of Neurology, Massachusetts General Hospital, Harvard Medical School, Boston, Massachusetts, USA. Let's break down its key contributions. Amyotrophic lateral sclerosis and frontotemporal dementia are devastating neurodegenerative diseases, and the most common genetic cause is a repeat expansion in the C9ORF72 gene. We know that this expansion leads to toxic byproducts, but it's been unclear whether the problem lies with the repeat RNAs themselves or with toxic proteins produced through a process called RNA translation. This study aimed to specifically address this question. To investigate the distinct roles of repeat RNAs and DPRs, the researchers used a clever genetic strategy. They engineered C9ORF72 mice and patient-derived neurons by mutating a specific codon that initiates DPR production. This mutation effectively silenced DPR synthesis while leaving the repeat RNAs untouched. They then observed the impact of this targeted intervention on various disease markers and outcomes. The results were striking. In the C9ORF72 mice, blocking RNA translation led to a significant alleviation of behavioral deficits and pathological abnormalities, including motor neuron loss and neuroinflammation. Importantly, this occurred even though the repeat RNAs continued to accumulate. Furthermore, base editing in patient-derived neurons also improved molecular phenotypes and survival, strongly supporting the therapeutic potential of targeting DPR production. This study provides compelling evidence that targeting the production of dipeptide repeat proteins, rather than the repeat RNAs themselves, holds significant therapeutic promise for C9ORF72-associated ALS and FTD. By successfully separating the effects of these two toxic species, the findings pave the way for developing novel treatments. The next crucial step will be to translate these findings into clinically viable therapies that can safely and effectively inhibit RNA translation in patients. Up next, our fifth paper out of 5, Recapitulation of Plaque Formation, Tau Pathology, and Neurodegeneration in a Human 3D Matrix Model of Alzheimer's Disease by Matthias Hebisch and Oliver Brussel from Institute of Reconstructive Neurobiology, University of Bonn Medical Faculty, and University Hospital Bonn, 53127 Bonn, Germany. Let's break down its key contributions. Alzheimer's disease is a devastating neurodegenerative disorder, and creating accurate in vitro models is crucial for research. Traditional cell cultures often fall short in replicating the complex pathology seen in patients. This study sought to overcome these limitations by developing a novel 3D human cell model. The researchers engineered human stem cells to controllably overexpress Alzheimer's-related genes within a 3D matrix. This system allowed for the induction of disease pathology using doxycycline. They employed various techniques, including advanced imaging and biochemical assays, to analyze the resulting cellular changes and confirm the recapitulation of key AD hallmarks. The results were striking. The model showed a significant 5-fold increase in the A-beta 42/40 ratio and a 12-fold increase in extracellular A-beta deposits upon induction. Crucially, these deposits exhibited characteristics of amyloid plaques, and the model also recapitulated tau pathology and signs of neurodegeneration, confirming its validity. This study presents a significant advancement in Alzheimer's disease modeling. The developed 3D system offers a more authentic representation of AD pathology, paving the way for better understanding disease progression and testing new therapeutic strategies. Its modularity also opens doors for future research, potentially leading to more effective treatments. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.